Cytology is the branch of science, is a branch of science or biology that is involved with the study of cell. Okay, that is involved with the study of cell. Cytology or cell biology is one of the same thing. So they are involved with the study of cell, types of cell, structure of cell, function performed by cell, and interaction between two cells. Cell biology deals with the study of cell, its structure, its function, shape, size, their interaction with other cells. Okay, cell biology is also called as cytology and is derived from Greek word kaitos, which means vessel. Okay, it is derived from Greek word kaitos, which means vessel. Cell is kind of vessel only in which all the metabolic activities of body perform. Okay? Cell is kind of a vessel only in which all the metabolic activities of the body takes place. So cell is called as fundamental unit of life because every single species on earth is composed of a cell, whether it is composed of only single cell or multi-cell. But every single species on this earth is composed of cells. It is called as fundamental unit of life. It is the smallest unit of life. Just like to build a wall, we need one brick. Okay, we need a brick and number of bricks together joined to form a wall. Okay, number of bricks together joined to form a wall. Similarly, cell is required for the formation of an organism. The number of cells combined together to form an organism. Number of cells combined together to form an organism. Here brick is a unit of wall. Here cell is a unit of life. Cell is a Latin word that means small room. It is like a room in which all the metabolic activities, all the important functions of life takes place. The cell was first discovered in the year 1665 by the English philosopher and architect called as Robert Hooke. The first time cell was discovered by Robert Hooke. The first time cell was discovered by Robert Hooke. So Robert Hooke discovered cell for the first time. Okay, he discovered hollow cavities like compartment. He discovered hollow cavities like compartment in a very thin slice of cork cell. Under his own designed microscope. At that time, compound microscope and powerful microscopes was not there. So he designed his own microscope to see hollow cavities like compartments in a very thin slice of cork cell. Okay, he took a slice of cork cell. And in this cork cell, he discovered hollow cavities like compartment. He took the cork cell of oak tree. That is Quercus plant. So he noticed this type of structure, honeycomb-like structure. This kind of structure he noticed when he observed that thin slice of cork cell. Okay? So this honeycomb-like cavities, honeycomb-like compartments were called by him cellulae. He called this compartment as cellulae and later cell. But the interesting thing is that he observed dead cells. Okay? He discovered dead cells that were not living cells because on this cell a suberin was deposited. On this cell there was some deposition. Okay, these were dead cells due to the presence of some depositions over this cell. So Robert Hooke observed dead cells. He was the first person to observe these cells, but dead cells were dead cells, and he observed cells in a thin slice of cork cell of oak tree. It is Quercus plant. Is it clear?
Ma'am.
Then comes the Anton van Leeuwen. He is also known as father of microbiology. He observed for the first time living cells. So whenever you are attempting the question, read it carefully. What is being asked? If in question it is asked that who discovered the cell for the first time, it is Robert Hooke. Who discovered the dead cell? It is Robert Hooke. But if it is asked that who discovered the first living cell, then it is Anton van Leeuwen, who is also known as father of microbiology. So he observed for the first time living cell. Okay. He took Pond scum and discovered single-celled organisms. That were actually moving. He called them animalcule, means animal-like, because they were moving. And if they are moving, it means they are living. Okay. He took pond scum and he observed that pond scum under a powerful microscope designed by him only. Okay. He designed a powerful microscope and noticed that there are some single-celled organisms that were moving, and they call them as animalcules, means animal life. Okay, clear? Then some timeline that in 1665 Robert Hooke discovered cell. Then first living cell was discovered by Anton von Leeuwenhoek. Okay. Then nucleus. We were going to talk about nucleus also. But 1883 nucleus was discovered by Robert Brown. Cell was discovered by Robert Hooke. It is Robert Brown who discovered nucleus. Flex Dujardin discovered that in cells some fluid content is also present. Then Sheldon discovered that all plants are made up of cells. Then Purkinje, he came and called that fluid as protoplasm. And you should know that protoplasm is the living material of cell. Protoplasm is the living material of cell. Okay, means in dead cell protoplasm is degenerated. Protoplasm is degenerated. So all living cells consist of a fluid which is called as protoplasm, which is a living material of cell. And this protoplasm term was coined by Purkinje. Is it clear? What is protoplasm?
Yes.
Then this Sheldon proposed that plants are made up of cells. Then with Sheldon, there was another scientist known as Schwann who said that all animals are made up of cells. Okay, this Sheldon and Schwann together gave a cell theory. Okay, Sheldon and Schwann together gave a cell theory. And now, what is cell theory? That all plants are made up of cells and all animals are made up of cells. Then there's Carl Brown who said that cell is the basic unit of life. And I explained you that for the formation of a wall, basic unit is brick, but for the formation of a life, the basic unit is cell. And there is one person who added one more point to the cell theory, who is known as Rudolf Virchow. He said that all cells arise from pre-existing cells. Okay, omni cellula e cellula. It means all cells arise from pre-existing cells. There is a saying known as omni cellula e cellula, which means all cells arise from pre-existing cells. This was the third point of the cell theory added by Rudolf Virchow. Is it clear? There is one more scientist known as Fontana discovered nucleolus in skin cell of eel. So nucleolus is one more cell organelle present in the nucleus. So Fontana discovered nucleolus for the first time in the skin cell of eel. So cell is the structural and functional unit of life. Why structural? Because all living organisms are made up of cells. Okay, and why functional? Because all metabolic activities takes place inside cell. So that's why cell is known as structure and functional unit of life and also the building block of life. Okay, next comes to cell theory, which was given by Sheldon and Schwann. Okay. Cell theory was given by Sheldon and Schwann. Sheldon was a botanist. Who is botanist? Who is botanist? Yes, who is botanist? Say something. Am I audible to you? Who is botanist?
Study about plant.
Yes, those who study about plants, and zoologists, those who study about animals. So they said that all plants and animals are made up of cells. The first point of cell theory. Second point, cell is structural and functional unit of life. Third point, cells arise from pre-existing cells. And this point was added by Rudolf Virchow. Is it clear?
Yes, ma'am.
Now, types of cells. We have two types of cells, prokaryotic cells and eukaryotic cells. Pro means primitive. Whereas karyotic means karyon. And karyon means nucleus. So, prokaryotic cells consist of primitive nucleus. Primitive nucleus means nucleus without any nuclear membrane. Nucleus without nuclear membrane means no proper nucleus is there. And just imagine that nucleus is like a center of a cell. It is like a center of a cell. It is like a brain of a cell. It's just like a brain of cell. Okay, means all the activities of cells, majorly takes place inside nucleus. Even DNA is inside nucleus. So primitive prokaryotic cells are primitive cells that contain no proper nucleus. Because nucleus without nuclear membrane means not a proper nucleus. All the contents of the nucleus will be present outside the nucleus. Means this is a cell and all the contents of nucleus is present like this because there is no proper membrane. That is called primitive nucleus. Whereas U means true and chaotic means again nucleus. So in this true nucleus is present. Proper nucleus is present. So that is with nuclear membrane. Okay. Prokaryotic and eukaryotic is clear. In prokaryotic, nucleus is without nuclear membrane and eukaryotic cells, nucleus with nuclear membrane. Is it clear?
Yes, ma'am.
प्रोकैरियोटिक में जो न्यूक्लियस होगा उसके पास कोई मेम्ब्रेन नहीं होगी, तो जितना भी न्यूक्लियस का मटेरियल होगा वो डायरेक्टली सेल में इस तरह से पड़ा होगा. बट यूकैरियोटिक में प्रॉपर न्यूक्लियर मेम्ब्रेन होगी एंड inside that this content of the nucleus will be present. This is a cell and this is nucleus. Proper nuclear membrane will be there. So pro means old or primitive and cryon means nucleus, and the history of evolution of prokaryotic cell is 3.5 billion years old, and prokaryotic cells the best example is bacteria. Okay, prokaryotic cell the best example is bacteria, and you know that how important bacteria are for us. For example, bacteria are present in curd. Which bacteria is present in curd? Lactobacillus bacteria. So these bacteria are responsible for converting milk into curd. They are responsible for converting milk into curd. Okay, bacteria are present inside our stomach also. They are important in industry for fermentation process, for research process. Okay, for example, lactobacillus bacteria, streptococcus bacteria. Different types of bacteria are there that are very much essential in different processes. Okay, in different forms, they are very important for us. Okay, and their history can be traced back 3.5 billion years old. So, what is actually there in prokaryotic cells? In prokaryotic cells, cell membrane bounded organelles are absent. Cell membrane bounded organelles are absent. Example, there will be no mitochondria, there will be no lysosome, there will be no endoplasmic reticulum, okay? Then chloroplast, nucleolus. So this all things are absent because this all contains cell membrane. So there is no cell membrane bound organelle present in prokaryotic cell. Okay, there is no cell membrane bounded organelles in prokaryotic cells. Whereas in eukaryotic cells, proper nuclear membrane will be there. Okay, cell membrane bounded organelles will be there. Okay, so eukaryotic cells are advanced. They are present in your plant, animal, fungi. Okay, so all the higher cells are eukaryotic cells. Is it clear? What is prokaryotic and eukaryotic? Is it clear or not? So difference between pro and eu. The cell size in prokaryotic is generally 1 to 10 micrometer. So in eukaryotic, it is 5 to 100 micrometer. Nuclear region is called as nucleoid because it is not surrounded by nuclear membrane. Nuclear region is called as nucleoid. Instead of nucleus, it is called as nucleoid because it is not surrounded by cell nuclear membrane. But here, nuclear material is surrounded by nuclear membrane. Here only single chromosome is present. Here, more than one chromosome is present. For example, 46 in human. Nucleolus is absent. Nucleolus is present. Membrane-bound cell organelles are absent. Membrane-bound cell organelles are present. Cell division by fission or budding. Cell division by mitotic or meiotic division. What is your fission or budding? Fission means breakdown. So one cell divide and into two cells. That is called as fission. Simple. Okay. One cell divide into two cells. It is called fission. And then again, this cell will divide and form two more cells. So this will continue. This is called fission. Splitting of cell. Okay. Splitting of cell. Whereas budding, suppose this is your organism. It will develop a bud. This bud will separate and form new organism. Okay. So budding and fission. Fission means splitting of one cell into two cells. Okay. Splitting of cell. It is called as fission. Whereas budding, a bud is generated. This bud will grow, mature, and will separate from your parent and grow into new individual. Is it clear? Fission and budding is clear.
Yes, ma'am.
Whereas mitotic and meiosis, what is that? One is mitosis or mitotic, and one is meiotic or meiosis. One of the same thing. So in mitotic See, cell is there. One cell contains 46 chromosomes in humans, I'm talking. Okay. So this cell will divide into two cells, but these cells will contain 46 and 46 chromosomes. Again, they will divide into two cells, but they will contain 46 and 46 chromosomes only. So it is kind of equational division. It is kind of equational division, and this takes place in all cells except your sex cell. Okay, this takes place in all cells called as somatic cells except your sex cells. Okay, this takes place in all cells, that is somatic or vegetative cells, except sex. In this, the number of chromosomes will remain same. For example, in your skin, in your heart cells, in your kidney cells, in your brain cells, mitosis is there, because when a baby is born, zygote is there. Zygote undergo mitosis division, and a full born organism is there. Okay, so at that time mitosis is there. In mitosis, the number of chromosomes remains the same, whereas when we talk about meiosis, it is called as reductional division. It is called as reductional division. Means that suppose this is a cell with 46 chromosomes. Now the two cells that are formed, they were having 23 and 23 chromosomes. Half the number of chromosomes. Okay. And this is important in sex cells, that is during your gamete formation. Remember, I told you law of segregation. Remember that 23 we will get from father, 23
Yes,
will
yes.
from mother. So at that time, meiosis is taking place, and this is important to maintain the chromosome number. Difference in mitosis and meiosis is clear?
Yes, ma'am.
Sure.
Yes, ma'am. Can you use white pen?
Oh, sorry. Is it clear?
Yes, ma'am.
Oh. Now, unicellular organism. Uni means single to single cell organism. Means organism made up of only one cell. Organism made up of only one cell is known as single cellular organism. Example, amoeba is there, paramecium is there, euglena is there. Okay, different types of organisms are there that are made up of only one cell. In that one cell only, all the activities are taking place, whether digestion, respiration, whatever activities are there are taking place in one single cell. So it is one single cell. You cannot see it with your naked eyes. If amoeba is present, euglena is present in your water bodies, so you are not able to see it with your eyes because they are so small. They are made up of only one cell, and you cannot see cell with your naked eye. You need a microscope to look this create. Clear?
Yes, ma'am.
Whereas multicellular organism made up of more than one cell is called as multicellular organism. Example, fungi, plant, animals. Okay, organism that is made up of more than one cell. So let's come to cell size. Cell size, variable size of cells are there. Variable sizes of cells are there. The smallest cell is mycoplasma or PPLO, pleuropneumonia-like organism. Pleuropneumonia-like organism. Okay, PPLO. Mycoplasma, these are cells of mycoplasma. They are the smallest cell on the earth. The size of this cell range from 0.1 to 0.5 micrometer. They are so small. Whereas largest cell that you can see with your eyes also, it is cell of unfertilized ostrich egg. Okay. Ostrich egg is one single cell. Ostrich egg is one single cell. It is the largest cell which you can see even with your eyes. Okay.
Yes, ma'am.
The size of it 15 to 18 centimeter wide. Okay, 15 or 18 centimeter, 15 centimeter length and 18 centimeter. This much large cell is there, known as ostrich. So when we talk about human, the smallest cell in human is sperm, largest cell is ovum, and the longest cell is nerve cell. In human, the smallest cell is sperm 5 micrometer, largest cell 120 micrometer, and the longest cell is your nerve cell. It is 90 to 100 centimeter, or you can say 1 meter. It is 100 centimeter. Okay, the size of this sperm nerve cell is about 90 to 100 centimeter or 1 meter. And you know the size of red blood cell, it is 7 micrometer. The size of red blood cell is 7 micrometer. See the variation of size of cells in humans. Also. Now shape of cells. The cells vary in shape also. And this variation depends upon the function of the cell, what kind of function they are going to perform. According to this, they are of different shapes. Some animals can change their shape. Some cells like single cell organism, euglena, paramecium, they can even change their shape. Most of the cells have a fixed shape. Okay, some cells like euglena and amoeba, they can change their shape. For example, white blood cells in human, which are responsible for immune system, for fighting the infection. Okay, they can change their shape according to the requirement of the body. Okay, according to situation, that to which kind of pathogen, to which kind of bacteria, virus they are dealing with. According to the situation, they change their shape. But most of the cells have a fixed shape. For example, RBCs, red blood cells, they are circular biconcave like this, and this shape is because they have to travel from one part of the body to the other part of the body. So due to this shape, they can easily pass from small capillaries also. When we talk about nerve cells, they are branched because they have to conduct impulses from one point to the another. So they are branched to conduct impulses from one point to another. Whereas when we talk about W bases, white blood cells, they change their shape because they have to fight with pathogens, bacteria, viruses. So they change their shape so that they can eat that microorganisms that enter our body. So they are of different shape. Is it clear? Is it clear? बोलो कुछ, तुम लोग बोलते क्यों नहीं हो? हैं? सदमा लग जाता है बीच में कोई? हाँ.
Ma'am,
ये बीच बीच में सो जाते हो? हाँ बोलो मैम क्या? What?
can you repeat, ma'am? Network.
Which one?
The last point we were saying, I couldn't hear because of the network.
Okay, about W bases.
Yes, I guess, ma'am.
Okay, I'll repeat this slide. I said that the red blood cells, they are of circular biconcave shape like this because they have to travel from one point of the body to the other, because they have to circulate. They have to carry oxygen from one cell to other cell. So due to this shape, they can easily pass through small capillaries or the small veins. They can travel through the small capillaries because of this shape. Whereas nerve cells, their shape is this branch shape. Branch shape is there of nerve cells. Because why? Because they have to conduct impulses from one point to the other. So due to their function, they have this branch shape. Whereas W bases, white blood cells, they have to fight with infection. They have to fight with bacteria, viruses that are entering into our body. They have to eat that bacteria, viruses that entering into our body, due to which they change their shape according to the pathogen entering into our body. जिस टाइप का पैथोजन होगा उससे फाइट करने के लिए W basis अपनी शेप को चेंज कर. ओके.
Yes, ma'am.
And I told you the example of prokaryotic cell is a bacteria. So see the bacteria structure. There is a capsule as the outer layer. Okay. Then we have inner layer of cell wall, and the third layer is plasma membrane. This together, three layers, plasma membrane, then cell wall and capsule is called as cell envelope. This is for protection. Okay. For movement of substances in and out of the cell. Okay, for movement of substances in and out of the cell. Okay, for protection as well as for movement of substances in and out of the cells. So there are three layers: cell membrane or plasma membrane, cell wall is there, and outer layer for extra protection there is a capsule. Okay, then in ribosomes are for protein synthesis. Ribosomes are responsible for protein synthesis. Okay, in bacteria, ribosomes is present, that is 70S type of ribosome. There are different types of ribosomes. So in bacteria, ribosomes are present, and that ribosomes are 70S ribosome. Then flagella is there. Flagella helps in locomotion, to move from one place to another. And this ground substance that is in blue color, it is called as cytoplasm, in which all other materials are present. Then villi is there. Villi is responsible for sexual contact. No sexual organ is there, just they come close together, they form a bridge in between them, and they divide. So no proper sexual reproduction is there, just a organ is there called as villi that helps the transportation of genetic material from one bacteria to the other bacteria. Then DNA, no proper nucleus is there, then it is known as nucleoid. I told you, when there is no proper nuclear membrane, no nuclear membrane is there, DNA is freely lying inside the cytoplasm. Okay, DNA is freely lying inside the cytoplasm. No nucleus is there, then it is known as nucleus. And one more thing is there, plasmid. Plasmid is extra chromosomal material. Plasmid is extra chromosomal material. Okay? DNA is there, chromosome is there, but extra chromosomal material which is present only in bacteria, that is known as plasmid. It is extra chromosomal material that helps in reproduction only. Okay? Cell division. Plasmid helps in cell division. Is it clear? Structure is clear?
Yes.
So I told you capsule, which is extra protective layer present in your bacteria. It is made up of polysaccharide. Poly means many and saccharide means sugar. Saccharide word is used for sugars. Okay. So this capsule is made up of polysaccharide capsule. It is polysaccharide capsule means made up of carbohydrates. It is made up of sugars. Okay. So their function is to give protection to the bacteria from drying out, from engulfment, because bigger organisms can eat bacteria. So this capsule give protection to bacteria from drying out, means to lose water, from losing water, and from engulfment by bigger organisms, because bacteria are very prone to phagocytosis. Phagocytosis means eating of bacteria, eating by other organisms. Other organisms can eat bacteria. That is called phagocytosis and cell. The capsule provide an extra layer of protection. This is capsule. Inside the capsule, there is cell wall and plasma membrane. Okay? So I told you cell envelope consists of three parts: capsule, cell wall, and cell membrane. Cell wall of this bacteria is very rigid, okay, stiff, made up of peptidoglycan. Peptidoglycan means protein. Peptido word is for protein and glycan word for sugar. Okay, peptidoglycan means protein and sugar which surrounds the bacteria, and there is another membrane known as cell membrane. Okay, there is another membrane inside the cell wall which is known as cell membrane. Is it clear? So three layers are there: cell membrane, cell wall, and capsule for the protection and the functioning of the bacteria. And the cell wall of the bacteria is made up of your peptidoglycan, or you can say as polysaccharide also. Then comes plasmid. I told you in bacteria there is DNA, but there are extra chromosomal part is also there called as plasmid. So plasmid is called as extra chromosomal material which is circular and present outside your actual DNA. Whereas mesosomes helps in mesosomes like this. This is your bacteria. So this layer of bacteria sometimes show folding in inner layer. Okay, this folding is known as mesosome. So mesosomes are nothing but they are the foldings of plasma membrane. Foldings of the plasma membrane. They help in respiration. Okay, they help in respiration. They help in the duplication of the cell. Cell division में भी ये help करते हैं. They help in respiration. They help in cell division. Mesosomes are nothing but they are the extensions of the plasma membrane or foldings of the plasma membrane. Plasma membrane or cell membrane, one of the same thing. Clear?
Yes.
Is it clear? Then three more structures are there: fimbriae, pili, and flagella. Flagella, I told you, it is for locomotion, for the movement. Okay, flagella is made up of flagellin protein. Okay, fimbriae is for attachment, to attach, because when bacteria enters in our body, they need to attach themselves with the tissues or cells of our body, or bacteria are attached over our skin. They are attached to some other bacteria also. So for attachment, they are having fimbriae. Fimbriae is made up of fimbrine protein. Now pili is there. Pili is made up of pillin protein, and pili is responsible for conjugation. Okay, it means transfer of genetic material from one bacteria to other bacteria. Transfer of genetic material, DNA, from one bacteria to other bacteria. It is with the help of pili. Okay, made up of pillin protein. Is it clear, these three structures?
Yes, ma'am.
And say pili are longer than fimbriae. The size of pili is bigger than the size of fimbriae. These three are made up of proteins. So now comes to eukaryotic cell. In eukaryotic cells, we have different types of cells: plant cells, animal cell, fungi is there. Okay, protozoans are there. So different types of cells are there in eukaryotic cells. So I am talking about, when we are talking about eukaryotic cells, they are plant cells, animal cells, fungi cells are made up of. Some protozoans are there, not all, but some protozoans are there that are eukaryotic. Okay. So when we talk about animal or plant cell, to see the diagram, from here you can make some differences. So animal cell size is 30 10 to 30 micrometers in length, whereas in plant cell size is 10 to 100 micrometers in length. See the shape, typically round or irregular, but see here shape is rectangular or cubic. In plant cell vacuoles, see vacuoles, they are small. This vacuole is there, this vacuole is there, this vacuole is there. But in animal cell, see size of vacuole is very small and numerous. But in here vacuoles are larger as compared to animal cell. Okay, as compared to cells of animal, plant cell contain large vacuole. Okay, and mostly one central vacuole is there. Okay, in plants there is no animals, no cell wall is there. Cell wall is absent, but in plants, cell wall is present. So some differences are clear: size, shape, vacuole, and cell wall. Some differences are clear, and
Yes,
here
ma'am.
one more extra structure is there: centriole. Here no centriole is there. Centriole is for cell division. Centriole is for cell division. So in plants, centriole is present. In animals, centriole is present. In plants, no centriole is there. So one more difference is created. So let's see different. Plant cell usually they are larger than animal cell. Cell wall is present, but here cell wall is absent. Plasmodesmata is present. Plasmodesmata is a connection between two cells. Plasmodesmata is a connection between two cells, but here plasmodesmata is absent. Chloroplast is present because they are responsible for photosynthesis, but animals are not undergoing photosynthesis. So chloroplast absent. Vacuole, large and permanent vacuole is there. Small and temporary vacuoles are there. The tonoplast, what is tonoplast? Tonoplast is a layer of, or you can say that wall of vacuole. Okay, so tonoplast is present around vacuole. Tonoplast is centrioles absent except some cells of your lower plant, but mostly it is absent. Okay, but centrioles are present for cell division. Nucleus usually present at the along the periphery of the cell, at near the walls of cell, but nucleus usually at the center of the cell. Lysosomes are present, but they are very rare. Lysosomes are present. We are going to discuss about lysosomes, vacuole, everything. So in plants, lysosomes are very rare, but in animals, lysosomes are properly present. In plants, the storage material is starch. Okay, glucose is stored in the form of starch. Glucose is stored in the form of starch, but in our body, glucose is stored in the form of glycogen in leaf. Okay? Glycogen is stored form of glucose in humans or in animals. Plants and animal cell is clear. Sure. Now cell wall is clear. I think cell wall can be found in plants, fungi, prokaryotic cells, but absent in animals and protozoa. Cell wall are absent in animals and protozoa. Cell wall gives additional protection. Okay, to help to hold that pressure, because plants, fungi, they have to expand when water enters their cell, and while expanding they should not burst. जब वो expand करते हैं वो फट ना जाए, burst ना हो जाए. So there is cell wall to give them extra layer of protection during expansion or during stress condition. Okay, cell wall help them to rigid. Cell walls are very rigid, so they help them to locate at one position, especially in plants, because plants are standing, wind is coming, they have to undergo stress condition to locate, to be remain at their position. अपने position पे बने रहने के लिए इनको extra layer की जरूरत पड़ती है, और वो extra layer क्या है? Cell. In plants, cell wall is made up of cellulose. This is the major content. Okay, with cellulose, hemicelluloses and pectin also there, but the major component of cell wall in plants is cellulose. Plant cell wall is made up of cellulose, whereas fungal cell wall is made up of chitin. Okay, fungal cell wall is made up of chitin. It is also known as fungal cellulose, whereas in plants the major component is cellulose, and in your bacteria, cell wall is made up of peptidoglycan. Okay. In industrial uses, cellulose is very much important, and we obtain cellulose from wood, from cotton, and this cellulose is used to produce textile, paper. Okay, so we obtain cellulose from wood pulp and cotton, and this is used for the production of textiles and paper. Even the exoskeleton of arthropods like cockroaches, insects, they are made up of chitin. The बाहर वाला जो skeleton होता है cockroach का, insects का, that is made up of chitin. Exoskeleton means outer one. There are some diatoms whose cell wall is made up of silicic acid or silica acid. Okay, the bacterial cell is made up of peptido gland. In plant cell wall, there are three types of cell walls in plant. Three layers are there. Primary layer is there, then middle lamella and secondary layers. In plants, three layers are there. Primary cell wall is there, middle lamella is there, and secondary walls are there. Okay, secondary walls can be 1, 2, 3, 4, 5. Okay, according to the age of the plant, according to the thickness of the plant, they are secondary walls. Okay, so see secondary wall 1, secondary wall 2, secondary wall 3. Okay, then there is primary wall of pink color and outside middle lamella. Middle lamella is basically your cementing material. Middle lamella is basically your cementing layer that actually you connect one cell to other cell. Okay, this middle lamella is a cementing material that connect one cell to other cell. So middle lamella, it is called cementing layer that join two adjacent cells. For example, this is one cell and this is another cell. So these two cells are joined by this middle lamella. Then there will be primary layer and inside there will be secondary layer. So outside middle lamella, primary layer and then secondary. Okay. Middle lamella is basically made up of calcium and magnesium pectate. Middle lamella is basically made up of calcium and magnesium pectate. And you should know that when fruits ripe, ripening of fruit is due to dissolution of this pectate. Okay, ripening of fruits is due to dissolution of pectate in middle lamella. जब middle lamella में ये pectate dissolve होता है, then fruit की ripening होती है. Fruits ripe due to dissolution of pectate in middle lamella. Is it clear? Then primary and secondary cell walls, they are made up of cellulose. Okay, primary and secondary cell walls, they are made up of cellulose. And primary cell walls, they are found in all young growing cells. They are found in all young growing cells. And whereas secondary cell walls, they are found in cells that have stopped growth. Okay, they are found in cells that have stopped growth. Okay, primary cell walls, they are found in all young growing cells, whereas your secondary cell wall is found in cells that have stopped growth. Is it clear? Primary cell wall is made of cellulose, and where secondary walls, they are inner to primary wall. Okay, and they are made up of dead cells because they are found in those cells that have stopped growth. Secondary cell wall, they help in the thickness of the plant cell. Okay, secondary cell walls, they help in the thickness of the plant cell. So this is about cell wall. Any doubt till here? It is clear? Yes, ma'am. Yes, ma'am. So